Dear students, we are excited to invite you to join an enriching webinar on enzyme action mechanism and kinetics, where you will gain valuable insights into the fascinating world of enzymes and their essential role in biochemical reactions. After completing this webinar, participants will be able to understand the specificity of enzymes, substrate interactions, discuss the relationships between enzyme structure and its active site, and explain the derivation of the Maclaurin equation and investigate the factors that influence the enzyme activity. I encourage each of you to actively participate in discussions, ask thought-provoking questions, and your curiosity and enthusiasm will make this session more impactful and enriching for everyone. We extend our deepest gratitude to our distinguished speaker, Professor Neera Kapoor Garu, Department of Life Sciences, Indira Gandhi National Open University, New Delhi. Thank you, madam, for accepting this our invitation. Now I will hand over the session to the speaker, Professor Neera Kapoor Garu, to deliver the lecture. Thank you, madam. Madam, sorry for the
Okay,
inconvenience.
audio.
Yes.
Good morning to all of you. Very warm welcome to all my learners, undergraduate and postgraduate learners. And I'm really thankful to Dr. Kishore for giving me a chance to discuss on a very important topic of enzymes. So can I share the PPT now?
Yes, ma'am.
Okay. Is it visible? Hello?
No, madam. No, madam.
It is not visible?
Yes, ma'am.
It is. My PPT is visible?
Not visible, madam. Your PPT is not shared.
Not shared.
स्क्रीन ये तो पीपीटी वाला ये वाला है ये वाला है ये ये your screen sharing ठीक है
Yeah.
ये तो
Now it is okay?
Yes, madam. Now it is okay, madam.
Yes. It is fine now?
Yes, ma'am.
Yeah. So enzymes, as we know, that they play a very important role in metabolism, diagnosis, and therapeutics. Now, whenever you speak about enzymes are always there whenever we study in physiology or we study in biology or in cell biology. Because enzymes, they are biocatalysts and all the reactions, they are being catalyzed. So an enzyme, as we know the catalyst, what is a catalyst? Catalyst is any substance which activates or it decelerates any reaction without being used in the reaction itself. And the level of enzyme, they are of diagnostic importance. This is very important. In physiology, we know that in myocardial infarction or during the heart attack, there are some enzymes which increase. For example, the level of troponin, it increases during the whenever there is a heart attack, and this level increases within 3 to 6 hours. There is a means a sharp increase in this particular level of the enzyme. And there are the other enzymes also which also show increase during myocardial infarction, like creatine kinase. This is also an enzyme which increases there. So it is a good indicator in the diseases. Then enzyme, it can always also be used therapeutically, such as some of the digestive enzymes. They can be used in the treatments. Now, as I mentioned, the enzymes, they are proteins. They increase the rate of reaction by lowering the energy of activation. What is activation energy? That I will tell you, and we will discuss in detail. They catalyze nearly all the chemical reaction taking place in the cells of the body. You know, our body is engineered in such a way that many metabolic reactions are taking place, and several they are taking place spontaneously, simultaneously, and these enzymes, they are not altered or consumed during reaction, and they are reusable. Now, active site, it is very important. Enzyme molecules, they contain a special pocket or cleft, which is called the active site. As you can see, this is the active site. This is the substrate which enters the active site of the organism. Enzyme changes the shape slightly as substrate binds, and then this forms the ES complex. This is EP complex, that is enzyme product complex, and this is the product which is leaving the active site of organism of enzyme. Now, what is a apoenzyme and what is a holoenzyme? Enzyme, most of the enzymes, they are proteinaceous in nature, except for ribozymes, which are not proteinaceous in nature. Now, the enzyme, without its non-protein moiety, it is termed as apoenzyme, and it is inactive. Here you can see. And holoenzyme, this is an active enzyme with its non-protein component. So this is the apoenzyme, and this is the cofactor. We will also mention about cofactor later on. This is the substrate, and this holo means whole. So this is the whole enzyme which becomes the active. Apoenzyme, which is not active. Now, important terms to understand the biochemical natures. Cofactor, this is very important. Cofactor, it is a non-protein chemical compound that is bound either tightly or loosely to an enzyme, and this is required for catalysis. Now, the types of cofactors are coenzymes and prosthetic groups. Now, these are very important, and generally the difference between these two is asked in the examinations. Now, the coenzyme, this is also a non-protein component. Prosthetic group, this is also a non-protein component. But coenzyme, this is loosely bound to apoenzyme by a non-covalent bond. Whereas prosthetic group, it is bound tightly to the enzyme by the covalent bond. As we know that the covalent bonds, their interactions are very strong. Now, what are the examples of the coenzymes and prosthetic groups? Coenzymes, vitamins or compounds derived from vitamins. But remember here that all the vitamins, they are not coenzymes. Some vitamins, they are coenzymes. Now, the prosthetic groups, copper, heme, biotin, flavin, these are the examples of the prosthetic groups. Now, enzyme specificity, enzymes have varying degrees of specificity for substrates. They may recognize and catalyze single substrate, group of similar substrates, a particular type of bond. Now, the table, it shows the types of enzyme specificity, absolute, group, and linkage. Absolute reaction type, it catalyzes one type of reaction for a single substrate. For example, urease, it catalyzes only the hydrolysis of urea. Group catalyzes one type of reaction for similar substrates. First one was for single substrate, it is for similar substrates. For example, hexokinase. This is very important enzyme during the glycolysis. It adds a phosphate group to hexoses. Then linkage. It catalyzes one type of reaction for a specific type of bond. For example, chymotrypsin, it catalyzes the hydrolysis of peptide bonds. Now, activation energy, as I mentioned earlier, what exactly this is the initiation energy basically. Here we have given the example that how enzymes lower a reaction's activation energy. Here you can see that this reaction, it is without enzyme. This reaction, it is with enzyme. We know that lactose, it is complex polysaccharide, which is disaccharide, which is made up of glucose plus galactose. But when there is no enzyme, then just see how much pressure or energy is being utilized. And here you just see when the enzyme is there, then how it is being done so easily, because this enzyme, it lowers the activation energy. Now, activation energy is basically, it is a first push to start the reaction. This is what we call the kick, the inertia. This energy, that is called as the activation energy. Now, mechanism of action of enzyme, these are very important. Enzymes increase the reaction rates by decreasing the activation energy. Now, there are two models, lock and key model, induced fit model. Lock and key model, basically, this is an old model, and induced fit model, it is a new model. Now, in lock and key model, you can see the active site. This has a rigid shape. Only substrate with the matching shape can fit. Substrate is a key that fits the lock of the active site. Here you can see substrate is key, lock, this is enzyme. So if proper key it is there in the lock, only then it will open. So this explains the enzyme specificity. But now in the lock and key model, the active site has a rigid shape. Here you can see this is enzyme, this is substrate, this is active site. Only substrate with the matching shape it can fit. Substrate is a key that fits the lock of the active site. This is an older model and does not work for all the enzymes, because I told you there are multi-enzyme reactions. Many substrates are there, and a single reaction is also there where the multiple enzymes will be there. So this model does not fit there. So there is another model, that is induced fit model. In this, the active site, this is flexible. It is not rigid as it was in the lock and key model. Shapes of the enzyme, active site, substrate, they adjust to maximize the fit. So what happens? The catalysis, that is the main function, that is improved. There is a greater range of substrate specificity. This model is more consistent with the wider range of enzymes. Now, this is the enzyme substrate complex. E plus S, we know it is giving the ES. This is the complex. So then enzyme product complex is formed, that is EP. This is the transition state. Then EP, then this E will be there, enzyme, which will be reused, and the product is formed. So this is the particular mechanism which is taking place. This seems to be very simple, but this mechanism, it is not so simple. Here we are showing the simplified representations, but in body, so many reactions are happening that it becomes very difficult. So that I will discuss about the enzyme kinetics also in the later on in my presentation. Now, what affects the enzyme activity? Now, there are very important factors which affect the enzymes. First of all, environmental conditions. Second, cofactors and coenzymes. And third, enzyme inhibitors. I will mention in detail about how the inhibition, enzyme inhibition, it is being taken place in the body. Now, in environmental conditions, the first factor is extreme temperature. Extreme temperature are very dangerous because we know that enzymes are mostly proteins, and proteins, they get denatured or they get unfolded in the high temperatures. pH, most like that is 6 to 8 pH, near neutral. That is ideal for enzymes. But different enzymes, they work in the different conditions. Some enzymes, they work in the acidic conditions, and some they work in the alkaline conditions. Then substrate concentration, that is very important because enzyme acts on the substrate, and as a result, product is formed. So how much substrate concentration is there? Based on that, many equations, they are being generated, which I will mention later on. Now, cofactors and enzymes, I mentioned inorganic substances like zinc, iron, and vitamins. They are sometimes need for proper enzymatic activity. Iron must be present in the quaternary structure of hemoglobin in order for it to pick up oxygen. Now, environmental factors, you can see the optimum temperature. This is the temperature, human body temperature, 37 degree. This is the optimum temperature at which the enzyme reactions, they occur fastest. Now, pH also affects the rate of enzyme. Most of them, they function between the neutral pH, but some of the enzymes, for example, pepsin, this works in the acidic conditions, and the trypsin, it works in the alkaline conditions. Now, substrate concentration and reaction rate, this is very important. Now, here you can see that this is the concentration of substrate. In the unit of this is mole per liter. And here this is the V, or the initial reaction rate. You can see here that as we increasing the concentration of the substrate, the maximum velocity, or Vm, this is being attained here. Okay. And furthermore, if you are adding the enzyme, this is saturated, far more substrate than it can deal with. So this activity increases up to this, and then you can see a plateau. It is there because all of the enzyme, it is combined with the substrate. The rate of reaction increases as substrate concentration increases at constant enzyme concentration. This is very important. Maximum activity occurs when the enzyme is saturated, when all the enzymes are binding the substrate. That is the maximum, or the saturated stage. Now, the classification of enzymes, there are different types of enzymes and different types of reactions which are taking place. So these are the classes: oxidoreductase, transferase, hydrolase, lyase, isomerase, ligase. So whenever you are studying about different pathways, like glucose pathways, or the glycolysis, gluconeogenesis, citric acid cycle, and other mechanisms, and in the DNA synthesis also, DNA replication, protein synthesis, all these enzymes, they are being used there. So I will tell you about these enzymes. The International Union of Biochemistry, it is there. According to this, enzyme has two parts. First part is the name of the substrate on which it acts. Second is the reaction catalyzed by the enzyme. For example, lactate dehydrogenase. And generally, you will see that the suffix ase is being used there. And here, when the lactate is there, it means the lactate is a substrate on which the enzyme will function. And this is dehydrogenase. So here we have given the many examples. So we know what are the activity of the dehydrogenases. EC number. Enzymes are classified into 6 different groups. Nomenclature, it was determined by Enzyme Commission, and all the enzymes are assigned an EC number. The classification, it does not take into account amino acid sequence, protein structure, or chemical mechanism. This is not the thing. Okay. Now, EC numbers, they are 4 digits. For example, A, B, C, D, where A is the class, B is the subclass, C is the subclass, and D is the subclass. For example, alcohol here, NAD plus oxidoreductase EC number is 1.1.1. This you don't have to understand at this level, but this I have done. This just I have told you that this is being done. How the classification is being done. Our main emphasis is on the type of the enzymes. Oxidoreductase, they catalyze the transfer of hydrogen, you can see, or oxygen atoms or electrons from one substrate to another. These are the redox reductions. So electron donor, it is required to complete the reaction. Here you can see the reaction, and the examples are oxidases, dehydrogenases, and reductases. Then transferases. They catalyze the group transfer reactions, excluding oxidoreductase, which transfer hydrogen or oxygen, and they are EC1. These are of general form. Examples are transaminases, which transfer amino groups, kinases, which transfer the phosphate group. Hydrolases, they catalyze hydrolytic reactions, so water will be always there. So examples are lipases, esterases, amylases, peptidases. Lyases, these are very important enzymes. They catalyze the non-hydrolytic removal of functional groups from substrate, and they create a double bond. You can see here, it was a single bond here, but here there is a double bond is being formed. Or the reverse reaction can also take place, that is addition of functional groups across a double bond. Examples are decarboxylases, aldolases, dehydrases, deaminases, synthesis, etc. Now, isomerases. They catalyze isomerization reactions, including epimerizations and cis-trans isomerizations. Isomerases, that is cis-trans, or DL, that is dextro-levo, right side or left side, epimerases, which are called as. Ligases, these are very important. They catalyze the synthesis of various bonds, which are coupled with the breakdown of energy containing substrates, usually ATP.
Madam, sorry to disturb you, madam.
Yes.
Can you please share the PPT so we are unable to see, madam?
The PPT is not coming?
Yeah, yes, madam.
Achha, it
That
is.
bonds are, we are not identify that bonds, everything.
How the PPT it is. Till when it was coming, up to what point it was coming?
Actually, a few slides only we are, sir, madam, but then only we don't know.
Now this slide, it is not coming, this ligases.
Yes, ma'am.
This is there or it is not there?
It's not there, madam. It's not visible your slides.
But why it is showing here screen sharing?
Coming now.
Coming?
Yes, madam. Yes, madam. Okay, it's okay, madam. Thank you.
It is coming, no?
Yes, ma'am.
Yeah. So, for example, ligases, they are very important. That is, they are coupled with the breakdown of energy containing substrates. So, for example, during the synthesis of DNA, there are very small segments which are also called as the Okazaki fragments. And these Okazaki fragments, they are combined together with the help of ligases. So these are very important enzymes which take place in our body. So in this particular section, what I told you about what is the importance of the enzyme, how they act, what is the classification of the enzyme, what is the activation energy, what is the significance of activation energy, and what are the different models by which enzyme acts in our body. Now, this is very important, that is enzyme kinetics. This needs a lot of understanding also, because whatever enzymes they are means catalyzing the reactions in our body, but how the rate of reaction is being taking place, and what is the significance of the substrate concentration. And there are some equations also for this, which we will mention here. And then how the enzyme particular reaction, it is being inhibited by different substances. So this I will mention. Enzyme kinetic, it is a study of the chemical reaction. It is visible, it is coming the slide. Hello?
Yes, ma'am. Yes, ma'am.
Acha, okay. Enzyme kinetic is the study of the chemical reactions that are catalyzed by enzymes. What exactly happens in enzyme kinetic? That the reaction rate is measured and the effects of varying the condition of the reaction investigated. What we want to determine: maximum velocity, this is very important, substrate affinity, inhibitor and affinity. What it can tell us: flow through metabolic pathways, utilization of substrates, what we can do with this information, control and manipulate the metabolic events. Now, just see here, this is the substrate here, this is the substrate. This is the enzyme. So you can see here that with the enzyme, this substrate is there. This is the low substrate and this is the high substrate. So this is the E plus S with now this K, it is there. Then there is the ES and then there is the P plus E. Now, what is this Km? This is what exactly it is the Km. That is the substrate concentration at which the reaction rate, it is the half V max. That I will mention later in Michaelis mental equation. Basically, this is our initial rate period. How quickly the enzyme becomes saturated with the particular substrate and what is the maximum rate it can achieve. You will understand it here. This is the important equation, Michaelis mental equation. Now, this basically it is a traditional equation which we study for the enzyme substrate reactions. But in this equation also, there were some drawbacks they were observed. And as a result, some new equations they were derived from this. But this is the basic equation from which other equations are derived. Now here, each and every this thing we have to understand. Here V0, it is equal to V max into S, then Km plus S. Now, first we will see here this particular graph. This is the substrate concentration and this is the reaction rate. Okay. Now V0, it is the initial reaction velocity, and V max, here you can see this is the maximal velocity. This is the substrate concentration. So this particular equation is that V0 is V max into substrate concentration over Km plus S. Now what is the Km? V max, half of V max, this is the half of V max. Wherever half of V max it is reached here, that is the Km. So this particular concentration at which the reaction rate is half V max, this particular. This is the V max. So this is the Michaelis mental equation which shows that this is how the reaction is being taking place. This determines the rate of the reaction. How the rate of the reaction is being determined in our body. The main thing of Michaelis mental equation is that it is based on the steady state assumption. That where substrate concentration, it is greater than enzyme concentration, and at initial velocity rate, it is assumed that the changes in the concentration of the intermediate ES complex are very small. We know that when E and S is formed, intermediate ES complex is there, by which then EP is there, then E plus P are there. So it is assumed here that the changes in the concentration of the intermediate ES complex, they are very small. This is called as the steady state rate, and these kinetics are called as the steady state kinetics. It follows that the rate of ES formation will be equal to the rate of ES breakdown. I know you will find it quite difficult because there are some mathematical things also here, some chemical things are also here. But this will become more simple as I will tell you another reactions also. So here, this you have to understand, this is the basic reaction. This is the central reaction around which all other the reactions will be, they can be derived. Now, in Michaelis mental equation, it was found that it was useful for simple reactions, for single enzymes. But for multi-substrate enzymes, it was not found as ideal. And it was also not found ideal for product inhibition. So what it was done was another reciprocal plots they came. This was the Lineweaver work double reciprocal plots. It was found that this particular plot, it could overcome all the difficulties and drawbacks which were observed in the Michaelis mental equation. Now, it is difficult to determine the Vmax experimentally. In a lab, you can study, when you study the experiments and you do the experiment and you want to study the enzymes activity, then you can see. Okay, but in the body, many enzymes, they are simultaneously acting. Then it becomes very difficult to determine the Vmax. So what exactly it is done in Lineweaver? Then the equation, it can be transformed into for a straight line by taking the reciprocal of each side. Each side the reciprocal is taken. That is why it is called as the Lineweaver-Burke double reciprocal plots based on the name of the scientist. And you can see here, this is the reciprocal is taken. And as a result of this equation formed is that is y is equal to mx plus b. And a plot of this will give the straight line with slope of Km by Vmax and y-intercept of 1 by Vmax. Such a plot, it is known as the Lineweaver-Burke double reciprocal plot. Here you can see. This is the 1 by V. Here this is minus 1 by Km. This is the intercept on y-axis, which is equal to 1 by Vmax. This is the slope, this slope, that is Km over Vmax. The biggest advantage to using the reciprocal plot, it is more accurate determination of Vmax and hence Km. It is also useful in characterizing the effects of enzyme inhibitors. As I mentioned, that Michaelis-Menten, it cannot characterize the effect of enzyme inhibitors, but this can. And it can distinguish between different enzyme mechanisms. Now, the significance of Km. Km, it is very important. You have to remember that Km, it is the substrate concentration at which reaction rate is half, that is half Vmax. This will change based on the different rate constants and at which step this is the slowest. In the simplest assumption, the rate of ES breakdown to product, it is the rate determining step of the reaction. So K minus 1 is greater than K2 and Km is equal to K minus 1 upon K1. This relation is also called as the dissociation constant for the ES complex and can be used as a relative measure of affinity of a substrate for an enzyme. However, if K2 is greater than K minus 1 or K2 and K minus 1 are similar, then Km remains more complex and cannot be used as a measure of substrate affinity. The main gist is that you should know about Km, Vmax, half Vmax, how to plot the graph. That is the main thing. Generally, the questions that we ask in the examinations are that how do you derive the Lineweaver-Burke plot from this Michaelis-Menten equation? And how it is more important than this Michaelis-Menten? And what exactly is the Michaelis-Menten equation? So these are the important questions which are being asked on these particular topics. Uses of Km. This is very important. Km, it is a useful parameter for characterizing the number or types of substrates that a particular enzyme will utilize. It is also useful for comparing similar enzymes from different tissues or different organisms. In many of the biochemical metabolic pathways, Km of the rate limiting enzyme determines the amount of product and overall regulation of a given pathway. Now, clinically, Km comparisons, they are useful for evaluating the effect mutations have on protein function for some inherited genetic diseases. So you can see that Km comparisons, they are so important that you can relate how the mutations they have occurred and how they are affected the proteins in some of the genetic diseases. Now, Vmax is the rate at which large substrate is the rate at a large substrate concentration, the maximal rate. The values of Vmax will vary widely for different enzymes, and it can be used as an indicator of the enzyme catalytic efficiency. Now we come to very important category of enzymes, that is allostatic enzymes. Now, what are the allostatic enzymes? We know that in enzymes, there is active site for catalysis. But in these allostatic enzyme, there is one more site that is called as the allostatic site. It is for the regulation. Now, these sites, they are located in the distinct places of enzyme molecule. Now, molecules that affect activity of an allostatic enzyme, they bind to the allostatic site. And these molecules, they are called as the allostatic effector. Now, one of the important allostatic effector is oxygen. It is for hemoglobin. You know that for hemoglobin, oxygen, it is very important if it has to be carried in the blood. And one of the important allosteric inhibitor, that's its ATP for phosphofructokinase in glycolysis. So here there is the another site is there, means there are the two different sites. Allo means the different. Okay, so they are the two different sites in distinct places of the enzyme molecular. Here you can see these are the given, but I think the this diagram, it is not very clear, I think. But these are the allosteric enzymes. Then there are the isozymes. Iso means similar. Different proteins which catalyze the same reaction. It is very important. Different proteins which catalyze the same reaction, they are called as the isoenzymes or isozymes. They are multimeric proteins containing two or more different polypeptide chains. Isozymes which catalyze the same reaction, they can be located either in different places of one cell or in different tissues. Isozymes, they have tissue specificity. Now, isozymes, they are created by different genes. Okay, so they have different molecular shapes. In as their isozymes, a similar category is also there, which I have not mentioned here. There are the allozymes also. Their difference is that isozymes, they are created by different genes, but allozymes, they are created by same genes. Okay, so this is very important. Isozymes and allozymes. Now, isozymes, they have some examples like lactate dehydrogenase, creatine kinase. So there are different examples are there. Lactate dehydrogenase, I will mention here. These are the isozymes of lactate dehydrogenase. You can see here, this is the muscle type and this is the heart type. In total, there are the 5 isozymes that you can see here. And this is their chemical formula. Now, a very important thing, that is the inhibition. If you have to study inhibition, we have to know about all the Vmax, Km, whatever things we have studied so far. Only then we can study about the and understand about the enzyme inhibition. Now, there are 4 types of inhibition: competitive inhibition, non-competitive inhibition, uncompetitive inhibition, and irreversible inhibition. We will take it one by one. Now, in competitive inhibition, what happens here? Now, first of all, you see, now this is the enzyme, this is the substrate, and I, it is for the inhibitor. Now, in competitive inhibition, the inhibitor, it competes with the substrate for the same binding site. This I will tell you with this example. Here you can see. This is E, S, E, S, E plus P. Now here what happened? This binds to the free enzyme only. This is the I and this forms the EI. If inhibitor is there, it means now the reaction will be inhibited and so what will happen? The further product, it will not be formed. So it binds only to the free enzyme, not to the ES complex. Okay, because enzyme it is here also, but this particular inhibitor, it has a tendency that it will bind here only. So now this is the general Michaelis-Menten equation. Now here this app word is apparent. Okay. Now V max app S. Here this is the Km app plus S plus. This form of Michaelis-Menten equation can be used to understand how each type of inhibitor affects the reaction rate curve. So means when we have to study about the inhibition of the 4 types of inhibition that we are going to study now, then this particular reaction we have to keep in mind. This we have to understand and remember this particular equation. Now in competitive inhibition, only the apparent Km, it is affected. Vx remains unchanged by the presence of the inhibitor. Just see this one. This is the equation here. What we told here is that the inhibitor binds only to the free enzyme, not to the ES complex. Therefore, apparent Km, it is affected. Km app is more than Km, but Vmax remains unchanged by the presence of the inhibitor. Example of this is sulfanilamide. It is a competitive inhibitor. This one, this is the inhibitor of for this particular acid. So these are also known as the sulfur drugs. Okay, they were the first effective systemic antibacterial agents. What we want is that the product should be the folic acid. Okay, but now if this particular inhibitor is there, competitive inhibitor, this is the inhibitor for this particular enzyme. Okay, so what happens as a result? Folic acid formation, it will not take place. But this does not affect the human cells, because in human cells folic acid synthesis is not taking place. So this was the example of competitive inhibition. We have to remember here that only Km is affected, Vx remains unchanged. Non-competitive inhibition. In non-competitive inhibition, you can see that the inhibitor, it does not interfere with the substrate binding. Here, just see here. It does not interfere with the substrate binding. This will be more clear next here. Just see. Here, what happens that inhibitor binds enzyme, okay, whether it is in free state or whether it is in enzyme substrate state. Means it has to react against the enzyme in whatever state it is. Earlier, we have seen that it was only inhibiting the enzyme in the free state, in competitive. But in non-competitive, it is binding the enzyme in whatever state it is. So what will happen? Now, just see this particular graph. What happens here is now, the inhibitor, it is there. So they decrease the Vmax app. You can see here. This is the same. They don't affect the Km. Okay. Km is same. Km app and Km, they are same. But this particular Vmax, this is getting decreased by the inhibitor. So Vmax app, it is less than Vmax, and but Km app is equal to Km. So this is different from that of the competitive inhibitor. Why does Km app is equivalent to Km for non-competitive inhibition? You have seen here this particular graph. Keep this in mind. The inhibitor binds equally well to free enzyme and the ES complex, because it is binding to both. In the first stage, it was not binding. So what happens? It does not alter the apparent affinity of this enzyme for the substrate, because it is binding to both. So affinity, it is not being affected. That is why this Km app and Km, they are similar, but Vmax app, it is less than the Vmax. So this is non-competitive inhibitor. Now they decrease the apparent Vmax, but they do not alter the Km of the reaction. Now, uncompetitive inhibition. In uncompetitive inhibition, what happens? The inhibitor binds only to the ES complex, neither to enzyme, not to the substrate, only to the ES complex. Just see. Here, ESI, it is being formed. It does not bind to the free enzyme. So in the first case, it was free enzyme. In second state, we have studied in both E as well as in ES. But in third, in uncompetitive, this binds only to the ES complex. So what happens? This uncompetitive inhibitors, they decrease both Vmax app and the Km app. You can note here this. Here you can see that uncompetitive inhibitors at low concentrations, they have little effect on the reaction rate. Here you can see, this is Km app, this is Km. So Km app, it is less than Km, and Vmax app, it is also less than this. So both Vmax app and the Km app, they are getting decreased. Why? Because this particular stage, it is being inhibited. This was the transition stage. This is the main crucial stage from which E plus EP, they have to be formed. So this is affecting on this particular stage. That is why both the things, they are being decreased. Uncompetitive inhibitors decrease both the Vmax app and the Km app of the enzyme. They don't bind to the free enzyme, so there is no EI complex in the reaction mechanism. They decrease the apparent Km of the enzyme and decrease the Vmax of the reaction. Now, irreversible inhibition, which cannot be reversed. What happens here? The inhibitor binds to the enzyme irreversibly through formation of a covalent bond with the enzyme permanently, inactivating the enzyme. Just see here. This is the mechanism. So inhibitor, what happens? This inhibitor permanently inactivates the enzyme. When the enzyme is activated, it means its function is now over. So what happens? The net effect is to remove enzyme from the reaction. So what happens? Vmax decreases, but there is no effect on Km. Why? Because enzyme is being removed. I think this will make you more clear. Michaelis-Menten plot here for an irreversible inhibitor. Here you can see. Now, here, because we know that this is inhibitor, it is binding with the enzyme in the initial stage only, and it is inactivating the enzyme. That is the main thing, that it is binding, but it is inactivating also. That is why it is irreversible. So what happens? Now, this Vmax app, this is less than this Vmax, half Vmax, and this Km and Km app, they are same. So irreversible inhibitors, they decrease Vmax app but leave the apparent Km unchanged. Irreversible inhibitors, they differ from other types of inhibitors. Why? Because they covalently modify the enzyme. And as we know that the covalent bond is very strong, so there is a permanent inhibition of the enzyme activity. So these are the 4 types of inhibitors which I've told you, and these you can understand very well if you understand the Michaelis-Menten equation, because that is the base, that is the central point for the studying the kinetics. Okay. Now examples of irreversible inhibitors: diisopropyl phosphofluoridate. It is prototype for the nerve gas serine. It permanently inactivates serine proteases by forming a covalent bond with the active site serine. So this is the example of irreversible inhibitor. Then there are suicide inhibitors. They work by tricking the enzyme into activating the inhibitor. They are suicidal in their own way because they activate the inhibitor. So what happens? They did form the covalent bond with the enzyme, leading to the permanent inactivation. Okay, so this is basically by tricking the enzyme into activating the inhibitor. So this is the summary for the enzyme inhibition. Competitive inhibitor, non-competitive inhibitor. We know that competitive inhibitor, it binds to substrate binding site. It competes with substrate. The affinity of the substrate appears to be decreased when inhibitor is present. That is, Km app is more than Km. Non-competitive inhibitor, it binds to allosteric site, does not compete with the substrate for binding to the enzyme. The maximum velocity appears to be decreased in the presence of the inhibitor. That is, Vmax app is less than Vmax. And then it is uncompetitive inhibitor. It binds to the enzyme only after the substrate has bound. the affinity of the substrate appears to be increased and the maximum velocity appears to be decreased when inhibitor is present. That is, Km app is less than Km and Vmax app is less than Vmax. And then it is irreversible inhibitor which covalently modifies and permanently inactivates the enzyme. I think I could make this clear with this because I know this is a bit complicated topic, this inhibition. This competitive inhibitor, non-competitive inhibitor, uncompetitive inhibitor, irreversible inhibitor, and then in between I mentioned about the suicide inhibitors which work by tricking the enzyme into activating the inhibitor. Why we are called it suiciding? Because enzyme itself is activating the inhibitor. And this inhibitor then it is working like an irreversible one, which forms a covalent bond with the enzyme, which leads to the permanent inactivation. So these are the 4 types of these inhibitors which I have mentioned here. So this is what I have told you in the enzyme kinetics, that there are what exactly is the kinetic, what is the Michaelis-Menten equation, what is its significance, what are its drawbacks, then what are the Lineweaver-Burke double reciprocal plot, what are its advantages, then what are the allosteric enzymes, what are the isozymes, and then what are the different types of inhibitions, and how they are deviating from the Michaelis-Menten equation. So this is, I could tell in this particular time about the inhibition. And in the first part, I mentioned about the, it was a bit simpler thing like classification and the induced fit hypothesis, lock and key. This you study in your 10+2 system also. That was a comparatively easy, but the second part, it was comparatively a bit difficult. But I think I could make some impact and I could get at your level to make you understand about how enzymes function and in our body. So you must appreciate that enzyme generally we say that what enzymes can do. But I think after listening to this particular topic, you must appreciate that how enzymes they are taking place in and how they are acting on different reactions in our body. Thank you very much. Stop shooting. Yeah.
Yeah, thank you,
Of
madam,
course.
for
Yeah.
thank you for your valuable lecture on enzymes. It was highly informative and greatly beneficial for students and enhancing their understanding of enzymes mechanism. Students, now, if you have any doubts, you can discuss with our speaker.
I think the topic, it was quite this thing because many equations were there and many graphs were there. So, but I try to make it a very this thing so that students can understand.
Yes, ma'am. It's a wonderful lecture.
So if you have any this thing, whatever doubt you have regarding enzyme inhibition, you can tell. Somebody has written. You can please write specifically what is the specific doubt you were mentioning.
Yeah, Rohit, please, which doubt you have regarding enzyme inhibition, you can ask directly.
But in
Students,
mixed, what mixed
students,
what?
you can unmute yourself and you can ask your doubts.
Yeah.
Yes, ma'am. The doubt is one student asked that in competitive and non-competitive inhibition, we see distinct effects on Km and Vmax. But
Yeah.
in mixed inhibition, how do we interpret the changes in both Km and Vmax in terms of enzyme substrate, binding affinity, and catalytic efficiency? Madam, hello. Okay, students, we can you can clarify your doubts during the online classes and direct counseling classes also. Now, I cordially invite Dr. S. Antikumari Garu to deliver a vote of thanks.
Is it audible?
Yes, ma'am.
Okay. Good afternoon, one and all. On behalf of the Faculty of Science and Department of Zoology, it is an honor and privilege to extend my gratitude to our esteemed speaker, Professor Neera Kapoor Madam, for delivering such an enlightening and insightful session on enzymes, action mechanism, and kinetics. Madam, your lecture has deepened our knowledge on the enzyme structure, types of enzymes, its catalytic properties, enzyme substrate interactions, that is enzyme mechanism, enzyme kinetics, Michaelis-Menten equations, and the various factors that influence enzyme activity. Madam, your expertise and in-depth explanation has truly been an engaging and thought-provoking. The way you explain on the role of enzymes in metabolism, diagnosis, and therapeutics is really more interesting as well as more educative. I thank you, Madam, for inspiring us and especially our students to explore this field further. We deeply appreciate the time and effort you dedicated today to make this session both informative as well as interactive. Thank you very much, Madam. I would also like to express my gratitude and thank our Dean and Director Academic, Professor Pushpa Chakrapani Madam, for her motivation, encouragement, and support to conduct today's webinar. Thank you, Madam. I would also like to thank all our teaching as well as the non-teaching staff and my dear students for their support and cooperation. Special thanks to the computer department for the technical support. Once again, I thank one and all for their enthusiastic participation, patient hearing, and in making this session a memorable one. Thank you, one and all.
Yeah, dear students, congratulations for all. Anyhow, you are attending this good lecture, and this is very important for your theory exams as well as your competitive exams also. Uh, in next week also we have one more webinar, so I hope same active participation we are expecting. Thank you all.
Thank you, sir. Thank you.